All right, so now let's talk briefly about cell communication. Cell communication is essential for all multicellular organisms. It's usually accomplished by some type of chemical means. We're going to talk about some different kinds of cell signaling. First of all, direct is when the cells that are communicating or the organisms that are communicating have to come into direct contact with each other. This would be like across the gap junctions between animal cells, like in the heart, through the plasmodesmata that connect adjacent plant cells, or between a helper T cell and a macrophage during an immune response. Local signaling, on the other hand, happens when cells that are nearby each other but not touching release chemical messengers that diffuse through space and allow them to communicate with each other. A good example of this would be the communication between two neurons at a synapse. Long-distance communication, on the other hand, happens between cells that are separated by long distances or organisms that are separated by long distances. Typically, the two most important ways of doing long-distance communication are, number one, by hormones, which we know are chemical messengers that are released by an endocrine gland into the bloodstream. They travel through the blood to specific target cells that have receptors for them. And once they bind to those receptors, sort of like a key fits into a lock, they generate some kind of response in that target cell. Pheromones, on the other hand, are usually released from an organism's body through sweat or urine. They then get into the air. They can travel very long distances and send signals about either safety, direction, or reproduction and mating. So let's talk about signal transduction pathways next. So a signal transduction pathway is a group of molecules in a cell that work together to control some kind of cell function. So after the first molecule in the pathway receives a signal, it activates another molecule, which activates another one and another one and another one. So there's lots of steps involved. And one of the most important things, and one of the most important reasons for all these steps, is that at many of the steps, the signal gets amplified. It gets spread throughout the cell so that the response can be a large response, even though there might have only been one signaling molecule that started that chain reaction. So let's discuss the 3 stages of cell signaling. This is something that gets asked about on nearly every AP biology exam. The first step is reception. This is when the ligand or the signaling molecule binds to a specific receptor. They fit together sort of like a key fits into a lock. Most of the receptors extend out into this outside the cell. They're extracellular receptors because most signaling molecules are made out of proteins and they have a hard time getting into the cell. Once they get together, that receptor changes shape, particularly the part of the receptor that extends into the cytoplasm. And this triggers step 2 of the process, which is the transduction pathway. A key word to know is ligand. A ligand is another name for the signaling molecule. And again, once it binds to that receptor, kind of like a key fits into a lock, it causes that receptor to change shape, go through a conformational change, and that triggers step 2 of the process. It's also important to think about this bulleted item right here. Different ligands can initiate different responses, while the same ligand can initiate different responses in different types of cells, because those different types of cells have different signaling molecules or different molecules involved in their transduction pathways. So there are exceptions to the extracellular receptors we talked about a minute ago. A few types of signals are made out of lipids or steroid based. Those substances can penetrate the cell membrane pretty easily, the phospholipid bilayer. So they actually have receptors that are inside the cell. They're intracellular receptors. Steroids are a good example. Estrogen and testosterone, the two we talk about the most. So they bind with an internal receptor, an intracellular receptor. Together, that receptor and that steroid then go through the nuclear membrane. They bind to specific genes within the nucleus and essentially cause those genes to be transcribed. In effect, they're turning on the genes. They're acting as transcription factors. Because they can enter the cell and they bind to receptors on the inside, they cause responses that happen a little bit slower, but they're usually more sustained than those responses caused by protein-based signals that bind to extracellular receptors. So step 2 of this cell signaling process is transduction. This is when the signal gets spread throughout the cell. It gets amplified. And like I said before, this is a multi-step process where one molecule activates another and another and another and another and another. Let's look at a picture of this. So in this case, we have one ligand binding to one receptor. That receptor changes shape, activates a molecule called as protein kinase 1. This then phosphorylates, it gives a phosphate group to another protein kinase, here labeled protein kinase 2, that activates it. That activates another one by phosphorylation, which activates another one, until at the end, we get the enzyme or the protein activated that we really want to activate. Now, what's misleading about this diagram is it makes it look like protein kinase 1 activates only one protein kinase 2, and protein kinase 2 activates only one protein kinase 3. When in actuality, at each step, one of these may activate several of these. One of these may activate several of these. So at the end, many of these proteins get activated so that this one ligand that we had up here at the beginning can cause a very, very large response. And I'm talking about sometimes times the millions. So one ligand might cause millions and millions of these active proteins at the end of the signal transduction pathway. So the final stage of cell signaling is the response. So the response is the action the signal was meant to initiate or stop or regulate. It might be something like opening up an ion channel, like in a neuron. It might be the breakdown of a substance, like the breakdown of glycogen that occurs in the liver in response to adrenaline. It might be the turning on of an enzyme. It might be the synthesis of an enzyme or a protein. Or another way of saying that might be the turning on or turning off of a certain gene, some type of gene regulation process. So that's response. So next we're going